Hey everyone, Mr. Murphy here. I'm going to be going through some problems involving Punnett squares. You've been assigned a few problems of a couple different types, which are asking you to look at the probability of certain types of outcomes when you mix certain individuals who have particular traits. Now, I'm hoping that we remember at least a little bit of this from middle school, but it's okay if you don't, or it's okay if you didn't actually end up seeing this in middle school. But the cool thing about a Punnett square is that it's able to allow us to look at the traits of two parents and to make predictions about the outcome of their offspring or children. So in this case of this first problem on the worksheet, you'll see that it's looking at a green plant which has a genotype of big G, little g. What that represents is that that plant received two alleles, or two copies of the greenness allele from its parents, and it receives one dominant allele, that's the capital G, and one recessive allele, that's the lowercase g. Now in the case of greenness, with what Gregor Mendel was looking at with pea plants, a plant is either containing the dominant allele, capital G, which means that it will show the green phenotype or the physical characteristic of being green, or it contains the lowercase g, which is the recessive phenotype, which would show up as being yellow. Now, in this case, the first green plant is big G, little g. We call that heterozygous. It means it has one of each of the possible alleles. But because it has one dominant allele, it will appear as just green. It doesn't matter if it's big G, or big G, little g. You wouldn't be able to tell the difference, because as long as something has one dominant allele, what that means is that it will show that trait. So we have one green plant, and it's being crossed with a yellow plant. Now the only way in which a plant could show the yellow trait or the yellow phenotype is if it had two lowercase g's, because it doesn't have that dominant green allele, and so instead of showing up as green, it shows up as yellow. What a Punnett square allows us to do is to figure out the probability of the different outcomes from this cross. And by cross, we just mean that if you reproduce these two plants together, what are the possible outcomes of that process? So in this case, we have one plant with big G, one plant with little g, and so you set up a Punnett square. It's just a square, and that is a poorly drawn square, but that's okay. And I'm going to set up 4 boxes here. And the reason why I do that is because if we look at this first parent, big G, little g, I'm going to show that the possibilities are that that parent could pass on a big G to its offspring, or that parent could pass on a little g. So every individual has 2 alleles, but what they pass on to their young is only one of those alleles. And if you remember, we talked about meiosis and how the process of making these haploid cells, or these cells that only have one copy of these different genes. And the reason why is because, just as a reminder, and in the process of reproduction, you're combining these 2 haploid genes from 2 different individuals to make one new individual who has therefore 2 copies of the gene, one from each parent. So this green plant could pass on a big G allele or a little g. And it's kind of a 50-50 mix of which one they pass on. Meanwhile, the yellow plant is either going to pass on a little g or a little g. It's 50-50 which one they pass on, but it doesn't really matter because they only have 2 lowercase or recessive copies to pass on. So when we set this up like this, what it allows us to do is pull one allele from one parent and one allele from the other parent. So I take this first column in this first row, and I show that one possibility is a big G from the first parent and a little g from the second parent. Now another possibility is that we could have gotten a little g from the first parent and a little g from the second parent, and so I write that in as little g, and we could have gotten a big G and the second little g from the second parent, or we could have gotten a little g and the second little g from the second parent. So in the end, we have a 50-50 shot of whether we're going to end up with an offspring or a new individual who has a big G, or a little g. And so when we do that, we want to write that out as we have 2 out of 4 possibility of big G, little g, and a 2 out of 4 possibility of little g, little g. Now it's important that we also distinguish whether we're talking about genotype, which is this, the actual genes that it receives, or phenotype, which is the physical like outcome. So if we look at this, we can say, okay, we have 2 possibilities of big G, little g, 2 possibilities of little g, little g, but what that really means is that we have 2 out of 4 possibility of a green offspring, or a 2 out of 4 possibility of a yellow offspring. Now in this case we only have 2 different possibilities, right? Big G, little g, or little g, little g. But in certain circumstances, depending on how these things are mixed and what their traits are, we could end up with as many as 4 different outcomes of genotype. And that could mean that we have 3 out of 4 green and 1 out of 4 yellow. In certain circumstances you're going to get 4 out of 4 who are green, 4 out of 4 are yellow, but we want to set up the Punnett square to show us which one is which. And again, just as a quick recap, what I'm doing is I'm looking at the genotypes, and I have a 50-50 shot of getting big G, little g, because there's 2 of them, and a 50% chance of getting little g, because there's 2 of them. Then I look at the phenotypes and I say, well, big G, any big G, whether it's big G, or big G, little g, any big G means it's going to be green. And if I have 2 lowercase, then that means it's going to be yellow. So that's how we approach these types of questions and figure out what the probability is of the different outcomes. In this second set of questions, it's a little bit less spelled out for us, but the words here are important to telling us how to set up our Punnett square. So again, I'm going to set up a Punnett square just like we did in the first problem here. And in these second sets, it's really just a matter of sort of discerning from the text what we're dealing with. So this tells me that we have a homozygous dominant brown mouse, and it's crossed with a heterozygous brown mouse. And then it tells us that the other option is that tan is the recessive color. So because we're talking about brown versus tan, the normal naming structure, just like up here how big G was green and little g was yellow, so in this case, if we have a homozygous, which means 2 of the same dominant brown mouse, that's going to be big B, big B. And heterozygous means 1 of each, so a dominant and a recessive trait. So if we have a heterozygous brown mouse, that's going to be big B, little b, because heterozygous means it has 1 dominant allele and 1 recessive allele, and that lowercase b is going to represent the tan color. Now you might wonder, why don't we use T for that? And really the answer is because that would get a little bit confusing in terms of a big B versus a lowercase t. And there are circumstances, if we were to go deeper into this, there are circumstances where certain traits actually can sort of blend together and can cause these even more unique outcomes, in which case you would use different letters. But when we're just talking about dominant versus recessive, you typically want to follow the rule of whatever your dominant trait is, that's going to be your capital letter, and that's what's going to determine what letter you actually use. So because we're talking about brown, I decided to use B's. Could I use T's instead? Sure. I mean, it doesn't really matter as long as you're following like the set rules, you're going to get the same probability and the same outcome. But it gets confusing if you don't follow sort of that norm, because if you're using T's instead, it gets confusing, like a capital T means brown. So instead we follow the rule of using the dominant allele as the thing that defines what letter we use. So if I were to do this Punnett square here, I have a big B from one parent mouse, and I have a big B, lowercase b from the other. And when I fill in my Punnett square, again using the column and the row, sort of showing you how these two things come together, and then I use this column and this row, big B, this column and this row for big B, and big B, little b. Again, I've got a 50-50 shot of different genotypes, so I have big B is 2 out of 4, and I have big B, little b is 2 out of 4. But if you look at this, because all 4 possibilities have at least one capital letter, one dominant allele, we end up with 4 out of 4 of these mice are going to be brown. And the significance here is that 2 of the 4, well, we shouldn't say it's necessarily 2 out of 4, but 50% of the time, your brown mouse that is the result of this combination will actually be heterozygous, which means that like in a future generation, you could see that tan color come back, because if you have a big B, little b mouse, yes, it looks brown, and you wouldn't be able to tell it apart from any other brown mouse, but the genetics of that mouse means that it carries that tan allele. And so in a future generation, you could see that tanness kind of come back. And that was sort of the main point of what Gregor Mendel saw, was that even though all of the plants of his first generation were tall plants, in the next generation he saw the shortness come back. And that's because some or actually all of those plants, in the case of Gregor Mendel's experiment, actually carried the short allele. So the thing that's kind of profound about this is that, again, Gregor Mendel's doing this in the mid-1800s and recognizing that there is a way in which these genes are actually sort of hidden or recessive. They're not shown, but the organism still carries them. And again, I can't stress enough how sort of significant that was at that time, because people didn't know about DNA. People didn't know about genes. These weren't things that we actually had a way of seeing and a way of manipulating like we do today, but still was able to determine that the probability of this stuff was predictable. And that's kind of the cool thing about these Punnett squares. So as you go through these, you want to set these up using the information that's given, and then think about, first off, think about the possibilities of the genotype, and the genotype is the actual genes, the actual alleles, capital or lowercase. And then you want to think about the phenotype, which is what it physically looks like. And so I always think of that as phenotype, physical, genotype, genetics. And that's what you're asked to do in these problems. So as you go through these, just think about, based on what's given, how do you set up the Punnett square, and how do you find the probability of these two things? It might be a little bit tricky in some of the cases, but I think with some practice you'll get the hang of it pretty quickly. And hopefully, like I said at the beginning, hopefully this will sort of call back to what we learned in middle school about Punnett squares and genetics. But I'd like you to hopefully make some connections to the discussion we had about meiosis, and how meiosis is all about figuring out which of your genes you might pass on to future generations, because you have 2, but you're only going to pass on 1 of each of them. And if you think about it, it shows you why, every person is unique. And I don't mean that in like the motivational poster on the wall, everybody's unique, diversity, yay. I mean that in the sense that, if we're looking at just one gene, whether you are a pea plant who is green or yellow, there's 4 possibilities, right? Or in this case, it's 50-50, but still, there's multiple possibilities for that one gene. Now add on to that, how tall is the pea plant? What shape seed does that pea plant have? All these different physical characteristics, and you start adding them in and adding them in and adding them in, and suddenly you realize that there's literally millions of different possibilities, or actually in the case of, more complex organisms, where you're talking about many, many different types of traits that cause all your different physical structures, your cheekbones and your skin color and your eye color, and all these different things. It adds up to just, billions or trillions of possible outcomes, which is why, 2 people aren't randomly born like completely identical to each other, except for identical twins. But that's a whole different story of how that actually comes to be, and that's because they actually started as one organism that split and then stayed split. A different story. It's pretty cool how that works out. But again, the point of this is showing us how the probability of this stuff is predictable, trait by trait, and how the numbers actually become pretty astronomical when you start looking at many traits at the same time. So as you go through these, follow this process of setting up your Punnett square, filling in your letters, and then figuring out, first off, genotype, big G's and little g's, and whatever the letter is, what are the different possible outcomes, and then thinking about, okay, based on that, in this case, 2 out of 4 are going to be green, 2 out of 4 are going to be yellow. But down here we see the same sort of split, except all 4 are going to be brown because we have at least one capital letter in all 4 of them. So they're going to show that brownness phenotype. So hopefully that's helpful, and if you have any questions on this worksheet, certainly let me know. Thanks.